Good day one and all present in the conference. Today I am going to present one of the research investigation of final year students of Bannariyaman Institute of Technology, Sattamangalam, Tamil Nadu, India. The title of the paper is Investigation of Antibacterial Properties of Chitosan in Textiles. Now, in this paper, we will try to see that application of chitosans on textiles. Now, if you see the functional behavior of the textiles, and there is an increasing demand to create some additional properties to the fabric, and we do this by imparting some active ingredients to the textile material. Now, as a matter of fact, the natural fibers, when you are talking, some of the important properties can be improved, that is insect repellency, odor and antimicrobial properties. So, how to do that with chitosan? Let us see now. Now, functional properties of textiles, when you are telling, lot of biomolecules are there. So, those biomolecules can be imparted to the textile material to improve certain properties. So, chitosan is one of a very important biopolymer available in nature, and it is sustainable, biocompatible, less toxic and abundant polysaccharides after cellulose. And there is wide application in the field of food, cosmetics and pharmaceuticals. So, these are the shrimp as commonly known as, and chitosan can be produced out of that from this biomolecule. Now, extraction of chitin is very important when we are talking about this chitosan, and here we represent that how the chitosan cells can be biologically or chemically, this chitin extraction can be possible. Okay. So, there are different processes: washing, drying, crushing, then demineralization. Okay. So, in that fashion we get ultimately the chitin. Now, there is another process which ought to be carried out, that is the deacetylation of chitosan from chitin. So, for that partial deacetylation, removal of the acetyl group and alkaline and enzymatic methods, those have been employed here. So, basic reason is to extract the chitin from some cells. Those are the crustaceans cells waste coming out from this chitosan, and there are enzymatic and alkaline method, and then deacetylation process, and after that we get that one way the deacetylation and another way we get deacetivation. So, that is the chitosan we are getting. Now, FTIR analysis, if you see that this is the FTIR diagram we are getting, and different bands we are getting at 3447 centimeter inwards, and we are getting the carbonyl group at 1477 centimeter inwards, and degree of deacetylation we are getting 1655 and 3450 centimeter inwards. So, this is the FTIR. Now, GCMS, gas chromatography mass spectroscopy, if you see in that, this is the chromatogram we have got from this chitin, and then we have received a lot of around 14 different compounds have been identified into that. Acrylonitrile is one of them. Then there is methyl pyridazine. So, there are a lot of such chemicals which can be really analyzed through this GCMS analysis. Now, coming to the antibacterial activity of chitosan, Sek flux method, AATCC, TM100 methods are employed, and two bacteria that has been used, that is the A. aureus, that is a gram positive, and E. coli, that is a gram negative bacteria has been used, and for determination of antibacterial activity, Sek flux method and AATCC TM100 has been used in this process. Now, antibacterial activity of chitosan has been determined, and there is high demand for this, and this kind of compound, if we apply, then it replaces the toxicity and it is biodegradable. So, this is a natural biomolecule. So, that is the important advantage of chitosan and application on textiles. Now, importance is the effect of degree of deacetylations. So, this how the reduction of this A. aureus bacteria is that with the pad dye cure method has been employed, and degree of deacetylation, if we increase, then we can get the reduction of A. aureus bacteria. Then the molecular weight, if you see that when the molecular weight increases with this method, then reduction of this bacteria above 90% against E. coli, and reduction on A. aureus also around 90% into that when that molecular weight of chitosan, okay, this has been increased into that. Now, there is increase in add-on percentage when you are talking, increase in stiffness of the material, okay, moisture regain also increasing, and there is a decrease in little bit of tensile strength of the material. Now, when we are applying the citric acid in the pad dye cure method, there is a reduction again about almost 100% when treating with 7% of citric acid and 0.8% that weight of the bath chitosan. And when you are talking about this tensile and tear strain retention of the fabric is there, is better here, 80% reduction of the bacteria after 20 laundering cycles, and wrinkle resistance properties also we have observed into that. And coming to the wool fabric also, you can see that when we apply to this pad dry cure technique, there also we can see that there is almost 100% reduction of gram positive bacteria A. aureus. Okay. So, even the deodorant activity, higher deodorant activity 30 to 40%, and relatively high laundering durability also we are getting. So, agar oil diffusion method has been employed into that, and we got the zone of inhibition, and this is the diagram of different disc diffusion method, and we are getting certain results of chitosan 2%, 3%, then with glycerin, honey, and honey with glycerin. So, best result we are getting with 2% chitosan, honey and glycerin combinations. So, that is the way actually this chitosan different percentage and different combination we are getting the zone of inhibition, okay, around 11.20 in this particular experiment. And there are other applications in the textiles also is there. It is also used as a binder, okay, and this is also used in the application of effluent treatment, because large amount of organic chemical those can be absorbed by this kind of chitosan in the textiles. And there are different dye shades we can get it chitosan in combination with different other compounds, okay. So, this is also a good dye. So, improved dyeability can be obtained, shrink proofing and weightability of the wool fabric can be increased with this. And this is also used as a thickener, synthetic thickener, it can resist the dye affinity. So, multiple application of chitosans are there in the textiles, agriculture, water and in the food processing industry apart from the different textile applications. So, the conclusion of this study is that it is the second most abundant polysaccharides after cellulose on the earth. It is biodegradable, biocompatible and non-toxic in nature and used for pharmaceutical, biomedicines, agriculture, food, cosmetics, textiles, enzyme immobilization and even more. So, antimicrobial property, antioxidant property, anti-inflammatory properties are there. And it is eco-friendly. Now, crustacean cell wastes are still underutilized. Okay, so we have to use more and more of such kind of product which are naturally available and abundant in nature, so that we can effectively utilize their property as a biomolecule on different textile product. So, these are the different references which have been studied into that, and based on that this study was carried out in the Bannariyaman Institute of Technology, and our final year students were involved in this study. So, Thank you very much for listening to this presentation. Thank you all.